Good evening, everyone. I'm Corinne Bush, the Executive Vice President of the Board for Certification of Nutrition Specialists, the BCNS. The BCNS is proud to partner with the American Nutrition Association, the ANA, in this ANA lecture series. The ANA is very pleased to be hosting Dr. Jeffrey Bland tonight to discuss phytochemical influences on metabolism and genetic uniqueness. Let me take a moment to tell you about the ANA. We're a nonprofit organization which for over 40 years has been on a mission to promote optimal health through nutrition and wellness education. We educate the public and professionals in many ways, including hosting world-class nutrition programs such as this one. We encourage all of you to visit our website at theana.org for an abundance of nutrition information, resources, and upcoming webinars. Join the ANA as a member and help us continue to bring these nutrition luminaries to you. And now we're honored to introduce Dr. Jeffrey Bland. Dr. Bland has been an internationally recognized leader in the nutritional medicine field for over 35 years and is well known for his ability to synthesize complex scientific concepts in a manner that is both personable and accessible. A biochemist by training, Dr. Bland earned dual degrees in biology and chemistry from the University of California, Irvine, and completed his Ph.D. in organic chemistry and the University of Oregon. He is a fellow of both the American College of Nutrition and the Association for Clinical Biochemistry, and he is a certified nutrition specialist through the Board for Certification of Nutrition Specialists. Welcome, Dr. Bland.
Well, hi, everybody. Good afternoon. I really appreciate once again the opportunity to talk about something that's been a passion certainly for all of us, and I would say has occupied the last 4 decades of my life pretty intensively, and that is how does diet and nutrition play roles in health outcomes? And so I'd like to play off the extraordinary presentations you've heard earlier today from Dr. Ames and Dr. Bloomberg, and I think this is a good segue into another part of the portfolio of nutrients that really relate to this interrelationship between our genes that give rise to our function. And these are the phytochemicals that I'm going to be focusing on, and I think Jeff did a really nice job of laying the groundwork for some of the detail that I want to deeper drill as it relates to these phytochemicals and the question of deficiency versus insufficiency. So just as a quick reminder, if we look at nutrients from the broad perspective and ask what do they do in human physiology at the cellular level, this slide basically is a course in and of itself. I mean, one could expand this out and spend several hours going through each of these. So the essential nutrients, the multivitamin and minerals, are, as we know, cofactors that influence then cellular function by the way that they regulate enzyme activity. Dr. Ames has pioneered this work. I'll be having an opportunity to speak a little bit more about that with Dr. Bloomberg and speak about Dr. Ames's work tomorrow. Secondly, there are these conditionally essential nutrients. These are substances that are made by our bodies, like taurine, lipoic acid, coenzyme Q10, glutamine, carnitine, tetrahydrobiopterin, but they may not be made at the levels that are necessary for the person's function, so they become conditionally essential, if you understand what I mean, based upon the status of that patient or person. Third, there are these food and spice-derived phytochemicals, and I'm going to be speaking about these. And then there's accessory nutrients that are thrown into the mix now, like prebiotics and probiotics and fibers and glycolipids and phospholipids and polynucleotides and glycoproteins and peptides, bioactive peptides. So these are not normally in a nutrition textbook as essential nutrients, but they play roles in modulating biological function, and they are food-derived. And then there's certain metabolites that we know play roles in modulating human function that are not necessarily considered nutrients, but are found in foods, both plant and animal foods, galactose, alpha-ketoglutarate acid, succinate, pyruvate, and this list can be very long of different, what Linus Pauling called orthomolecular substances, right? Natural to the organism. So this pharmacopoeia that I've just described is broad, and it makes the PDR look pale in comparison when you put all these things together in terms of the bioactive substances that are found in foods. And the mechanisms by which they can influence physiology, again, this is a whole other course in nutritional biochemistry, but just to quickly remind you, the B vitamins are the cofactors for things like pyridoxal phosphate, flavonoid dinucleotide, FAD, or NAD, nicotine adenine nucleotide, cobalamin, tetrahydrofolic acid or 5-methyltetrahydrofolate. So these are cofactors that relate to the regulation of specific enzymes that then modulate metabolic function, basically. And it's cell-specific, tissue-specific, and it's genome-specific as it relates to the needs that these substances have for regulating function. Dr. Bloomberg already did a beautiful job of describing that. Then we have antioxidants. And when I first started in this field, my first papers as a professor, as a young professor in 1970-71, were on vitamin E, the tocopherol family. And I look back at that work and I think, boy, we've come a long way since then, because we recognize that these aren't just like substances that prevent rancidity, like you put an antioxidant in oil to prevent it from becoming rancid. These are actually modulators of cell signaling. And so they have very specific effects beyond that of just soaking up oxygen radicals or nitrogen radicals. They actually can regulate then redox potential, which is a very finely tuned component of cellular function that ties back to mitochondrial oxidative phosphorylation. It goes back into this discussion of bioenergetics. So there's more to antioxidants than just soaking up rancidity factors. Then we have precursors to hormones, and we think of neurotransmitters and prostanoids. So we know that the omega-3 and omega-6 families of fatty acids are prostanoid precursors, eicosanoid precursors. We also know that various amino acids, and ones that are common would be tryptophan with serotonin, or phenylalanine and tyrosine with a dopaminergic, a dopamine family of neurotransmitters. So we know that these are bioactive precursors of various substances. Arginine, a precursor then to nitric oxide in the vascular endothelium. So these are ways that we mechanistically can identify the activity of these. And again, as I said, this is a whole course in and of itself. But I want to move on into, once again, examining what Dr. Ames has brought to your attention, what Dr. Bloomberg has brought to your attention, what you're going to hear much more over the course of this meeting, the difference between deficiency and insufficiency. We can look at these, what are called marker diseases, scurvy, beriberi, pellagra, xerothalmy, and rickets, and identify those clinically pretty easily. But you can go through a practice in Western medicine and probably never see a classic scorbutic patient or a classic beriberic patient. Certainly in developing parts of the world, you might see this, and even in maybe some really undernourished areas in the United States, you might see this. But in general, this is a very uncommon thing to see these classic marker deficiency diseases. But it begs the question, and it's a question that you have already heard eloquently discussed previously, is there just an on-off switch that you are properly nourished with these nutrients, and then the next moment you are beriberic, or you're pelagric, or you're xerothalmic? Or is there a graded sense of metabolic disturbance, which is what Dr. Ames has spoke so eloquently about with the triage model, where the body is going to maintain the most pivotal functions by soaking nutrients into that area and leaving other areas in your physiology that are less capably supported. So you're going to get these early stage symptoms. And as you probably recognize, there have been many studies that go way back, in my view, into the 60s, showing that the first signs of B vitamin insufficiency, in children at least, are psychological in nature. They often are learning disabilities, sleep disorders, behavior disorders, attention disorders. So you start to see certain marker tissues being the first thresholds of insufficiency before you get to deficiency. So that's an interesting part of the model. And as you probably know, Robert Heaney, who won the McCollum Award in 2003, which is the most prestigious nutrition award given in the United States annually by the American College of Nutrition, American Society of Nutrition. In his address, his acceptance address, he defined something he called long latency deficiency diseases versus short latency deficiency diseases. It's a very powerful concept. Let me tell you what it means. He said that it's easy to define a short latency deficiency disease because you can take medical students and give them a fee and starve them for a while and they'll get scorbutic or they'll get beriberic. And so you can study in concentration camp victims or in people on short duration of deprivation, you can pick up these marker diseases. It's much more difficult, however, when you look at these long latency nutritional disorders. And what are they? Osteoporosis, right, is a good example of a long latency. So it's hard to know whether you have a calcium disturbance, a vitamin D disturbance, or other things that might be related to something that takes 30 years for it to to exhibit in the phenotype. And he goes on to say that if you look at long latency nutritional inadequacy diseases, the family of disorders under that are virtually every chronic disease, right? So it's really easy to study the short-term deficiency diseases. It's much more difficult to understand the long latency insufficiency diseases from an experimental design perspective. And that's what I really try to talk about. Some of you know in my book that was published last year is this whole concept of what is a disease. And I'm not going to go through this. I think disease is a marker for reimbursement, but we really, there's kind of an interesting, what I would call relative scaling of disease, because what is a disease today, based upon the ability to do CAT scanning or do various SPECT scans or to look at radioimmunoassay, may be very different than what we would have defined as a disease 50 years ago without those tools. So people feeling crummy stays constant. What we call it changes. And the reimbursement codes that are assigned to it change. So the question really is not how we build a reimbursement system, but how do we understand the etiology of the sequence of events that leads to lower human performance, i.e. function, that ultimately crosses a barrier that we can put a label on it called a disease. Now, think of ICD-9s, right? ICD-9s, 15,000 diagnoses, right? And ICD-10s just came out in October. And what do you now know? 55,000 diagnoses. Now, what do we know about disease? When we just tripled the number of diseases in the same human population that hasn't changed its genotype. Doesn't that tell us something? It means it's a slippery slope, that we don't really have a hard understanding of how this all fits together. And we're putting new names on things in order to hold on to them based upon our new lenses of observation. So with that, I want to talk about food specifically, because food is this complex matrix that has all this various stuff in it that I'm describing, all of which has biological activity in every human being and speaks to our genes every time we eat. Speaks to our genes. Now that's a powerful concept, different than what I learned in school, some now more years than I'd even hate to admit, that particular concept that food speaks to our genes, and it can speak in different languages and different tonalities. It can speak stridently, or it can speak smoothly with kind of karmic destiny of great love and bliss. It depends on how you select what you're going to put into the mouth that ultimately ends up, transmitting its information to every cell of your body. So with that as in mind, I'd like to give a moment of thanks, because some of these beautiful slides that I'm going to share with you are not mine. They were developed by Dr. Deanna Minnick, who you'll hear on the program later this week, who is a colleague of mine for many years in our research department. And she's an artist as well as a nutritional biochemist, so I want to give some appreciation for a few of these slides that came out of her beautiful artistry. So our genes haven't changed over the last many years substantially, but the environment in which the genes are embedded or are expressed has changed dramatically. We all know that. This is like a rule of reasonableness. So food is constantly communing with our genome, but our environment, our food environment, and the environment in which we eat our food, and the other stuff that we're exposed to with our food has changed dramatically over the last 30 to 40 years. So how does this information in food get communicated into our cells to create these functional outcomes? That's an interesting question, right? And so there are, on the surface of cells, these receptors. And these receptors pick up information like antennae of different substances that are floating around in our cells. I'm going to talk about some of these receptors, like the G protein coupled receptors in a moment, which are capable of picking up information from our food, odorants and colorants and flavoring agents and other bioactive ingredients in our foods that are phytochemical in nature, can signal through this complex network called the kinase network, of which there are in excess of over 500 different kinases that are these enzymes that regulate the pachinko game of life and then ultimately communicate the message from outside cells to the inside of the brain of our cells, which is where the genome is housed in the nucleus of a eukaryotic cell, and turn on specific portions of our book of life, or turn them off. This is called gene expression, and it relates to the activation of promoter regions of genes that regulate then, not one gene at a time, but regulate families of genes that orchestrate themselves into our phenome. Am I making sense here? Okay, so with that as a model, what about the role of phytochemicals in this communication process? We're learning much more about them. What makes plant foods so special is that there are more than 10,000 that chemists have defined, 10,000 phytochemicals in foods, all of which can have some symphonic orchestrated voice in the symphony of life as to how they interface themselves with various gene expression agents and cell membrane receptor sites and signaling processes. Now, this is wonderful news for plant scientists because this is so many more Ph.D.s in the future. It'll keep all sorts of people busy. Think of all the kinds of studies that now with the new tools that we have with the deciphering the human genome and to be able to measure these various cellular processes, these signaling processes, what we're going to learn and are starting to learn about individualized nutrition. That there is no such thing as a perfect diet for everyone. It does not exist. There are the individual relationships of diet to a person's own specific genotypic status. So what are these phytochemicals? You already heard about them, so let me just kind of give you some oversight of their particular families. So we got terpenoids, we got phytosterols, we got phenols, thiols, indoles, isoprenoids. You know from Dr. Ames's work, like lipoic acid and ubiquinone, which is CoQ10. These then go down into subfamilies like the flavonoids, and you've heard again some really eloquent discussion about the flavonoids in the various families, and then ultimately to the individual members of those, which have names like quercetin or hesperetin, or if you go up to like into the glucosinolates, then you've got things like sulforaphane, and you've got indole 3 carbinol, and you've got dienoyl methane, and there's, as I said, 10,000 different organic chemicals that are secondary metabolites from these plants that ultimately regulate then function. So the retinols and all these materials that are coloring agents in foods have a specific biological functions. Okay, so we eat less than a teaspoon a day, but they make a huge impact on our health. So if you look at the gross amount in a day consumption, we get maybe about a gram and a half of phytochemicals of all these families in the average diet. So that's a pretty small amount, and I'm going to talk a little bit about what does that mean, and do we need more, and what's the evidence for the value of certain of these families. So how do these phytochemicals then speak to our genes? That's a big question, right? That's like when I was a kid, this is before the age of semiconductors, we used to build as small kids these cans with strings, and then we would speak into the can, and you would have the voice go to the other end of the string to the other can. So that's a way of speaking. That's a pretty inefficient way of getting information across. So the cells have a much more efficient process. It's called intracellular signal transduction. Right? So somehow these signals from food are transduced into the nucleus of our cells where they can communicate to our genes in very specific ways. Now, what are those ways? I promise you I'm not going to go through all these, but I just want to tell you that there are a number of ways that have been identified by which this process of signal transduction can occur. It can go through regulating nuclear transcription factors. It can go through epigenetic mechanisms, through various response element effectors like the antioxidants response element or the metal response element. It can go through redox regulation at the mitochondria. Remember, the mitochondria has its own genetic information that's passed on by the mother. There's more than 50% of us that's in our mother, in our genes. We're more maternal. I always say that our biochemical energy metaphorically comes from our mothers. And so you've got that mitochondrial DNA that's regulating some effects, enzyme inhibitors, precursor to cellular mediators, and post-translational protein modification. All of these play roles in regulating cellular signal transduction. So if I was to then look at in a diagrammatic way, you've got stuff floating in your blood after you ate. It's going to be distributed. So let's say that you just had a glass of green tea or a cup of green tea. So you got epicalocatechin gallate that's floating around. EGCG is floating around in your blood. And there are receptors on the surface of cells that have the affinity for that molecule. They have the right shape. They do the right dance. So they start dancing together. That forms a vibrational wobble in the cell membrane that transduces through the membrane, that then influences cellular bound proteins that then signal through the kinase signaling system. This could be through like protein kinase C, and it goes down through phosphatidylinositol kinase and into glycogen 3 kinase, and eventually it speaks to the genes in a very specific way by modulating transcription factors. Transcription factors are the things that actually cross the nuclear membrane and light on specific regions of your genome when they're opened up so they can read that portion of your book of life. Now, this is a pretty neat process when you think about it. So eating is a lot more exciting than you might have thought of just satiety or, it was like fun to have that experience of eating that candy bar. You're doing all sorts of other things in terms of cell signaling at the cellular level. And one of the principal families of receptors, not the only one, but the one I'm going to focus on here for the moment, are called the G protein coupled receptors, or GPCRs. And there are several hundred of these that sit on the surface of various cell types that then pick up information from your outside environment. Like when you smell something, you've got those odorant molecules, and whatever it is, could be a wonderfully smelling monoterpenoid compound with limonene in it, for instance, and that limonene goes in and it sits on the surface of one of these GPCRs, and it signal transduces to depolarize a cell, and a nerve response happens, and you like the taste of lemons, right? You smell lemons. So these are very powerful signaling transduction agents that tie together various organ systems. They can tie together the nervous system with the immune system, with the endocrine system. So you're getting a variety of different orchestrated effects from a single molecule across these receptors. So a GPCR then picks up this information. It transduces then this altered membrane potential, activates certain kinases, signals to certain regions of the genes, and now you get a different message. That cell didn't change its genes, it just changed the way it's responding to its environment. Am I making sense here? Okay, so this is a very interesting part of the story. Over 100 known GPCRs are activated by light, odors, pheromones, hormones, neurotransmitters, peptides, omega-3 fatty acids, and specific phytochemicals. So let's take one way that we know how to activate our GPCRs. That's called taste. So you got these taste buds. You got bitter, salty, sweet, umami taste that are sitting on the surface of your tongue, and you taste foods, and it's going to send a signal that's ultimately going to end up at the lateral nucleus of your hypothalamus to say, I like this, or I don't like this, or this stimulates my appetite. But it does more than that, doesn't it? Because those receptors that are sitting on the surface of your tongue are chemically identical to receptors that are sitting within the duodenal mucosa and the L cells of the distal ileum, meaning your intestinal tract is tasting your food. You get what I'm saying? Now, why is that in interest? It's interest because I'm going to start with bitter, because many of these phytochemicals that I've been describing, if you take them, isolated, they're bitter. They give you the taste of bitter. So what about bitter? Is bitter better or is bitter worse? Well, it turns out that in the family of bitterness, that there are certain types of bitter sensins that stimulate not only on the tongue the taste of bitter, but stimulate these introendocrine hormone receptor sites that are in your gastrointestinal tract. Now, what do these introendocrine taste receptors in your intestinal tract do? They stimulate, through these taster 2 receptors, the transduction of signals to the genes of those intestinal cells that causes the release of what? Glucagon-like peptide 1. Have you ever heard of GLP-1? Sure you have. You know there are a whole series of drugs, right, to treat diabetes that are GLP-1 either agonists or they're inexaktide, that what they do is they either block the breakdown of GLP-1 or they activate GLP-1. These are used to treat diabetes, GLP-1 drugs. How and why? Because GLP-1 activates, it's insulotropic, it stimulates beta cells to secrete insulin, it stimulates peripheral cells to be sensitive to insulin, and it's anti-inflammatory. It increases adiponectin and it lowers inflammatory mediators. Now you're going to ask, well, what happens then if you feed these bitter tastants to individuals? Can you measure postprandially elevated GLP-1? Yes, you can. Do you hear where I'm going with this? I'm saying that the introendocrine response to your diet is signaled by phytochemicals that are present in certain food families that modulate the way your body is prepared for the receive of that message, of that meal. So if you eat foods that are completely devoid of these messaging substances that were historically found in foods that we evolved with, what do you think it is? You get a blank kind of not here kind of message. It's devoid of that messaging. You might get a lot of calories, and in fact, if it's just a sweet message, let's call it an empty sweet message, what do you think it's stimulating there? It's not stimulating the L cells, it's stimulating the M cells. And what do they secrete? Ghrelin, and they secrete resistin. What do you think resistin does? It does the opposite. It resists insulin sensitivity. You get where I'm going? So you're sending signals to your GI tract that are then modulating systemically how your body is prepared to receive the message of those foods. And these signaling agents are to a great extent found within the phytochemical family. So here's soy isoflavones, right? So what do? They activate the taster 2R14 receptor and R39 receptor. What does that do? That regulates then aspects of postprandial glucose control and hormone levels. So we have this signaling network that is orchestrating how our body is going to respond to that meal. So if in fact what I've told you is right, one of the principles that we know with GLP-1 drugs is that they cause weight loss, don't they? So now there's a whole approval by the FDA for a certain dose regime of GLP-1 drugs in weight loss programs. But would you be surprised if I told you then that individuals who have a greater acuity for bitter taste are individuals who have lower BMI? Because they're getting the benefit of the metabolic effects of these signaling agents on their cellular physiology, right? Now we call these incretins. And incretins like Byetta and Januvia, those are the drugs that are called incretin drugs, are the same GLP-1 stimulators that I'm talking about. So these endocrine hormones are regulating all sorts of aspects of our function. Obviously, this is a talk that could go on for a long time all by itself, so I'm just giving you a kind of taste of the concept. So GLP-1 regulates multiple systemic functions. Look at the various tissues that it influences gene expression. So in the liver, it increases glycogen storage and it lowers lipid deposition, right? So it resists non-alcoholic fatty liver disease or infiltration of liver steatosis. In the brain, it's a neuroprotectant. It increases neurogenesis and increases memory. In the heart, it has effects on myocardial contractibility. In the pancreas, it stimulates beta cells to secrete insulin. In skeletal muscle, it causes improved glucose uptake and mitochondrial oxidation in muscle. I mean, what I'm trying to say is this is a pleiotropic series of effects across different tissues from the same signaling agent, which is activated by the way we eat. Now, what happens if we change the way we eat? You change the whole orchestra. The composition may still be a Tchaikovsky suite, but all you have is a percussion session. So you got a bunch of drums playing. That's what happens when you feed the wrong information to an orchestrated suite that never got played. You hear where I'm going with this? So how about omega-3 fatty acids? Well, omega-3 fatty acids have been found to work through the G-protein coupled receptors, specific ones. They stimulate then specific types of promoter regions of genes to be activated to induce certain genes, which happen to be the cassette of genes that are associated with what? Anti-inflammation. So they turn down TNF-alpha expression and IL-6, and they have effects on other things like adiponectin that upregulate anti-inflammation. So you can see that there's a whole cell signaling effect of these fatty acids. So phytochemicals work in multiple ways. Some of them are direct and some of them are indirect. So let's talk about dietary fiber. There's been a huge controversy for years. Is fiber good? Is fiber bad? Should we eat sawdust or should we eat oat bran, or what's good? And so what we know is different fibers. These are non-digestible polysaccharides that are not digestible by the normal human amylase enzymes. That these have specific effects on regulating the microbiome. So different bugs that live in our gut prefer certain kinds of non-digestible fibers as their principal food for fermentation. So you can alter then the gut microbiome, and you'll hear much more about this in greater detail from an expert in a moment, but we do know that the microbiome is connected to dietary fiber. So what happens then if you start looking at the way that certain fibers can influence then the GPCR activation in the gastrointestinal mucosa? That's a Nature Medicine paper recently that's explored this, looking at what happens when certain dietary fibers are activating then certain GPCRs that then release signals from gene expression systemically that do what? They serve as anti-inflammatories. So would you be surprised, this is a functional medicine metaphor 1A, would you be surprised if I told you studies have now indicated that certain fibers are useful for preventing chronic obstructive pulmonary disease. Because you reduce then the activation of certain inflammatory signals that are associated with the lungs that have morphology, or let's call it homology, is probably a better word, homology in their gene expression patterns to that of what's going on by the fermentation of fibers in the gut and the signaling of the gut immune system, the so-called gastrointestinal associated lymphoid tissue. So this diagram I took right out of Nature Medicine magazine, which is like a functional medicine diagram that shows fiber connected to bugs, connected to gut function, connected to gut immune function, connected to dendritic cells in the bone, connected to the lungs, right? It's a system, it's a network. You can't look at it as a pathway, you have to look at it as a network. Well, we also know this holds true in epigenetics, and you've all been familiar with Randy Jirtle's work and his then postdoctoral student Bob Waterland that reported remarkably the story on the folic acid and vitamin B12 and the agouti mouse and all that interesting part of the story. But now we learn that there are certain phytochemicals beyond B12, folate, B6, and betaine that influence epigenetic methylation patterns as well. It's not just those methylating nutrients, that phytochemicals can do this too. So here are some examples of various phytochemicals that have been found to modulate methylation patterns of the promoter regions of various genes. I already talked about EGCG, that's from green tea. And genistein, I've already mentioned that. That comes from one of the isoflavones from soy. Organosulfur compounds that come out of garlic. And lycopene out of tomatoes are all known to have effects on epigenetic methylation of the genome, as are then the compounds below, phenyl isocyanate from brassica vegetables, curcumin, obviously turmeric, sulforaphane, again from Brussels sprouts, cabbage, broccoli, and resveratrol from peanut skins and grape skins. So all of these are known to be phytochemicals that influence then methylation patterns of the epigenome. So that leads us to sirtuins. You've all heard about sirtuin 1 and its influence on age-related phenomena. When you calorie restrict, you modulate sirtuin 1, and that has a salutary effect on aging in animals. In fact, you can extend life spans of animals by almost 40% in these calorie restriction experiments. This is a very, very interesting part of the gene expression profile that regulates a whole series of genes that ultimately control things like insulin signaling and inflammation and bioenergetics through AMP kinase. So here's kind of how this all works in a grand scheme of things. Calorie restriction influences sirtuins. Sirt1 then influences a variety of downstream signaling agents like the inflammatory agent NF-kappa B. It blocks that. That's a transcription factor for inflammation. FOXO, which has to do with the metabolism of fat, and PGC1 alpha, which interrelates with PPR gamma and insulin signaling and inflammation. So that's a positive effect. So what we're saying is these are protective agents against biological aging or chronic disease relationships. So is it just calorie restriction that does that? And the answer is no, that we see these same positive effects occurring in animals that are fed these phytochemicals that modulate epigenetic methylation patterns and can influence sirtuin regulation. Why do they influence sirtuin regulation? Because the methylation of the genome regulates its structure, its folding and unfolding structure that allows for accessibility of the reader enzymes that are going to transcribe certain genes. So if you keep Fort Knox closed, you can't read certain genes. And remember, methylation patterns do what? They suppress or inhibit certain gene expression patterns, whereas acetylation of genomes open the doors to be read, that story to be read. So you get a dynamic effect on the way your genome is exposing itself to be read, because you certainly are not reading all the books, all the chapters in your book of life in every cell all the time, or you'd be a mess. Because in your book of life, you've got the message for every cell type at every age you live. If it was all expressed continuously, you would definitely be a mess. So you can only read certain portions of your book of life, and that's part of the regulatory process that's controlled by the way we eat. So phytochemicals regulate certain detoxification functions. You know this very well. You're going to hear more from Dr. Minnick on this topic. We had the pleasure of meeting a number of years ago, about 20 years ago, Dr. Eleanor Rogan at University of Nebraska, who helped us understand the role that certain phytochemicals play in the metabolism of estrogen. And I can just say that you know that 17-beta-estradiol and estrone undergo hydroxylation reactions. They can be hydroxylated at the 2, 4, or 16 position. These estrogen metabolites have different effects. We consider the 4 hydroxyestrogens to undergo odd oxidation into the 4 quinones, which are clastogenic. So they are associated in men with prostate cancer and in women with breast and endometrial cancer. So you don't want to make a lot of 4-hydroxy compounds. You want to push them into the 2-hydroxylated. And what do we know that does that? The thing that upregulates the cytochrome P51A2 is indole-3 carbinol, right? Which then activates a favorable detoxification of estrogen. So the 4s are the ones that are not so good. As I said, they're reactive to DNA. We want to get more in the 2s. So the 2s can then be worked on by catechol-methyltransferase, and what happens then is the 2-hydroxys get methylated to form the 2-methoxy, which is an anti-estrogen. So if you think of 17-beta-estradiol as being the estrogen accelerator of cellular mitotic activity, then you think of the 2-methoxy estradiol as being the brake, right? And you need to have your foot on both pedals, both the accelerator and the brake. Whether you're a man or a woman, principally estrogen's higher in women, we know, but men have estrogen in their prostates. And if they get a lot of these funny estrogens, that is also, as has been shown by Rogan and her colleagues, very strongly associated with prostate cancer. So sisters get breast cancer, brothers get prostate cancer, right? With similar defects in the way they metabolize these compounds. And it can be modulated by the way we eat. The cruciferous vegetables contain the phytochemicals that help to regulate gene expression of these modifiable, or these upregulatable enzymes that convert the estrogens into the less active forms. So here are a review paper that really talked about these bioactive phytochemicals that modulate estrogen through the effects they have either at the receptor site, the ER alpha or the ER beta, or the estrogen metabolism. And that the range of these is quite, the range of the plant foods is quite broad, from turmeric to chili peppers with capsaicin, to ginger with gingerol, to green tea with EGCG, soybeans we already talked about, lycopene, cabbage with I3C, broccoli with sulforaphane. All of these then help to modulate the way these signals of estrogen travel through the body. And there are clinical trials, as you know, done in administering I3C in a purified form. This would be about 400 milligrams a day, is equivalent to about 3 portions, normal portion sizes of broccoli a day, to give you an idea. And when you do that, you give it to women in a placebo-controlled trial that have PIN, which is a pre-cervical dysplasia condition. You see return of their cervical cells back to normal cytology. They revert back to normal architecture. So this has actually been demonstrated in a couple of interesting placebo-controlled trials in women with PIN. Let me give you another example of this. This is one that we were actively involved in, and I think published over 30 papers in this area, and I won't bore you with all the papers, but we looked at 2 indigenous families of phytochemicals and their synergy, how they work together. One were the so-called isohumulones that were derived from Humulus lupulus, which is hops. As you know, it's a bittering agent in beer, and it's a grass food ingredient. And the other is a series of anthocyanidins from the indigenous African tree, the bark of the tree called Acacia nilotica. Those combination of those 2 families of chemicals, the isoalpha acids from hops that are represented in the structures A, and the specific anthocyanins found from the nilotica bark, as shown in structure B, when you put them together in different ratios and feed them to animals, you get different effects. And I'm not going to go into this, but there is a symphony that's translated in different ways depending upon the ratios of one family to the other family. So you can modulate lipid metabolism, you can modulate glucose metabolism and insulin sensitivity in different ways depending upon the ratio of those compounds in the food. And we published a whole series of papers on this. I won't bore you, but looking both in cell models and in animal models and even in human intervention trials. And some of you know this was a paper that I'm actually very proud of because it may be one of the few papers in the literature that did a high-quality control diet, which was the Mediterranean diet, and then another group of randomized patients with a similar intake. These were metabolic syndrome patients. Another group of patients of the same type who got the same exact diet, except they got supplemental levels of these phytochemicals. So everything was the same calories, same protein, same carbohydrate, same fat. The only difference was the addition of these phytochemicals. Now, what did you find? What did we find? We found that because both groups were put on this Mediterranean diet, they both had improvement in all of their parameters, lipids, insulin, inflammatory markers, all of that improved. But there was on average a 30 to 40% improvement beyond that of the diet alone by giving supplemental levels of these phytochemicals. So it's a pretty outstanding example of how you go from deficiency to sufficiency to improving function even beyond sufficiency, finding the right kind of dose response. So phytochemicals prevent an inflammation of chronic disease. They interface with the gut, and you're going to hear much more about the microbiome in a moment, but we now know that the microbiome will convert some of these phytochemicals into secondary metabolites. One that's an example is Equol. You probably know a little bit about Equol. Equol is one of the soy-based phytochemicals that gets converted by microbiological fermentation in the gut by specific types of bacteria into an anti-estrogen and an anti-metastatic agent called Equol. And Equol has all sorts of effects in benign prostatic hypertrophy in males and in benign breast disease in lowering inflammation of these tissues. So that's not the phytochemical itself, that's the secondary metabolite in the gut by friendly bacteria that produce Equol. Are you getting where I'm going here? So it's a community. It takes a village to create this, this particular response. So small amounts of phytochemicals in the diet can have significant physiological influence. This is called a hormetic effect. It doesn't follow a normal dose-response relationship. Small doses can have unexpectedly high effects if they're delivered at the right time in the right place. Hormesis with these phytochemicals has been examined by a variety of investigators. One whose work I really greatly have a lot of respect for is Dr. David Sinclair at Harvard. He and I have done quite a bit of collaboration in looking at these synergies of, in this case, resveratrol, curcumin, and ECGC, and modulating these complex intercellular signaling pathways that cut across inflammation, mitochondrial bioenergetics, and insulin signaling. They all interrelate to the modulation with these phytochemicals. And it doesn't take a whole lot to do it, but it takes the right amount at the right time. So have we become chronically ill due to the fact we've taken a lot of these messaging substances out of our diet? That's a really interesting question. The average American eats 1.5 servings of vegetables and 1 serving of fruit a day, including French fries and ketchup. And so we only get about 11% of the people meeting their guidelines, as Dr. Bloomberg has already pointed out. So there is an increased need for the delivery of these favorable components of the diet to regulate cellular architecture and cellular orchestration. Again, this is just once again a review of what you'd already heard from Jeff. If you look at stroke, 26% less stroke with people who had more servings of fruits and vegetables. Or if you look at the intake on type 2 diabetes, vegetable intake and fruit intake on diabetes, with an increase of 1.15 servings a day, there was a 14% decrease in the incidence of type 2 diabetes in an epidemiological study. This is another study looking at green leafy vegetables specifically and their reduction of risk based upon the consumption patterns of the people with a higher intake had the higher incidence of diabetes. Here's the flavonoid. This is another examination of the same study that Jeff was talking about, high flavonoids with reduced cancer risk of all organs. And dementia, this is a series of papers. I only chose one here to illustrate the point. 41% reduction statistically in dementia with those that consume the higher quintile of intake of flavonoids. So these are substances, they're not vitamins, they're not minerals, they're not even essential. You know, if you go to a standard textbook of nutrition, they often occupy only like a couple of pages in the text because they were considered to be kind of like unimportant. They just came along for the ride. In fact, they might have even imparted bad food characteristics. So the processing industry, the food processing industry wanted to take them out because it would make the food taste more bland so they could put more sugar and salt in it and make it more saleable. So we've eliminated many of these phytochemicals just by the nature of processing. So lastly, I'll just say there are ways that one can actually assess the phytochemical composition of the diet by the so-called phytochemical index. And there are now published papers showing that people who consume diets that are high or in phytochemical index, this would be like an RDI for phytochemicals, that they have much better overall health and function. If we want to talk about healthy aging, once again, to just reinforce what Jeff Bloomberg was talking about. And these are the slides that I really want to give Dr. Minnick a lot of credit for. This is her beautiful artistry. So if you really look at the colors of the diet as being a watchword and a guide, and I'm not talking about synthetic colors, I'm talking about the machinery in the plants that produce these secondary metabolites called phytochemicals. So you have red phytochemicals like lycopene, anthocyanins, and astaxanthin, and they have their own unique effects upon biological function. Or you've got orange phytochemicals like beta-carotene and bioflavonoids that have a different role to play in how they modulate cellular signal transduction. Or you've got yellow-green chlorophyll-containing and lutein-like substances that have another series of effects on how they regulate function. You notice I'm talking about a variety of different plant foods here. It's important because you're getting all these different families under the rainbow. And lastly, blue-purple then has another series of effects on, and these coloring agents are markers for the phytochemicals that are in them. That's why I'm using the colors as a watchword. So there's a real easy takeaway message if you're eating white and gray and brown foods, or foods that have been colored synthetically to make them look like they mimic a phytochemical, you're on the wrong path, right? That's not sending the right signals to the genes. So, well, and it's white, obviously. You do have some white compounds like allicin in garlic. So I leave you with the concept that if you look at the green, red, white, blue, and orange foods, that we fall short based upon dietary surveys in considerable levels of percentage of the population, 69% in green, 70% in red, 88% in blue-purple. That means if you talk about the difference between deficiency, and by the way, there is no specific deficiency disease, marker disease, that can be identified for a phytochemical deficiency, right? So these are all insufficiencies that I'm talking about. These are long latency nutritional inadequacy diseases, to go back to Robert Heaney's concepts. So we have completely missed it. These are like a blank page in our learning that is now being filled in by this extraordinary breakthrough discoveries of how foods influence gene expression. Over the times that we eat, the days of the month, the months of the year, the years of the decade, we shape how our genes are expressed, and ultimately we become the phenotype of our diet. We look, feel, act, and think as we have been treated by our food. Thank you very much.